All right, great. Thank you so much for having me here. My throat was itchy when during the previous talk I got COVID and I was feeling it. Anyway, so in case I start to clear my throat, and you know why. I clearly recovered, right? So I look okay now. So anyway, so today I'm going to tell you about our work as part of the ENCODE consortium, and to continue from our effort during the ENCODE consortium. And today is going to be more like the evolutionary aspect of these regulatory elements that controls gene regulation. So just a little bit of motivation. So as you all know, 1.5% of the human genome encode for proteins. And we are really very interested in regulatory elements, which is, we think, the interesting bits of the non-coding genome. And the reason we're interested in regulatory elements is ultimately we're interested in understanding the variants that differ among our genomes. How are they related to our phenotypes? The interesting thing is these genetic variants are highly enriched in regulatory elements. So, and I have a pointer, so actually, is this one brighter? Okay. So we are interested in regulatory elements. So just very briefly, regulatory elements in ENCODE, we call them candidate cis-regulatory elements because these are defined using epigenetic data or transcription factor binding CHIP-seq data. So they're not like thoroughly tested. That's why they're called candidate. And very often they include promoters, which promotes the transcription of a gene by interacting with the proteins that will bind to the promoter or interact with the polymerase. And enhancers, they act at a distance by working through promoters. And they are silencers and they are insulators. So we commonly see these different categories of regulatory elements. And what I'm going to talk about today includes all of them. So today's talk is not going to go into like different types, but just as a whole, they're all in part of our consideration. So they are so interesting that there is a 2-decade ENCODE project that just ended earlier this year to chart all these functional elements in the genome. And I have an old picture before COVID, when people are still attending in-person conferences. We had one that was more virtual later on, but you get the picture. So ENCODE wanted to catalog functional elements in the human and the mouse genomes. And we were playing quite a bit of role in the data analysis center of this project. And so the project generated a ton of data, and all freely available at encodeproject.org. And we actually had quite a lot of fun analyzing the data. And for the talk of today, I'm going to mention a few data types. One is DNA-seq, which detects open chromatin regions in the genome. And they are CHIP-seq, chromatin immunoprecipitation, followed by sequencing. They can be for grabbing out the regions bound by transcription factors, abbreviated as TFs, or histone modifications. And there are a bunch of transcription data, RNA-seq, various types. And we worked hard on defining the registry of these cis-regulatory elements. So, so how do we define these cis-regulatory elements, candidate cis-regulatory elements? I started out by saying they're candidate because we use epigenetic data. So exactly what do we mean by using epigenetic data? So these are like elements that are nucleosome sized. They're around 300 base pairs long and very commonly devoid of a nucleosome. And instead of the nucleosome, they're bound by transcription factors. And very often they will be bound by multiple transcription factors, up to 10 sometimes. We actually have data to show that a distribution. So if you have antibody that specifically recognizes one specific kind of transcription factor, you can chip it and then find out where it binds. That's one way of detecting these cis-regulatory elements. And ENCODE has a bunch of these kind of data. Or they are very often flanked by nucleosomes that have a specific histomodification pattern. Itself is nucleosome free, but the neighboring nucleosomes have patterns. And you can also use antibody to grab down those histomodifications and then map and find out the region between them to get these CCREs. Or you can use assays like DNase, which detects open chromatin. It basically cuts open chromatin regions and then try to figure them out where they are and then map to the genome. So we integrated a bunch of these kind of data that's generated by ENCODE, as you can see here, in a specific locus of the SP1 gene. And we detect these regions, like 300 base pair long regions in the genome that correspond to promoters that are open chromatin and has histone mark, enhancers and insulators. They have a characteristic histone mark and binding by transcription factors such as CTCF, which is the insulator binding protein in mammals. So we detect these CCREs. So as part of ENCODE 3 effort, so actually a lot of work was done by Jill Moore and Michael Picaro and Henry Pratt here, and Henry is sitting right there. So if you have any questions that I cannot answer, go ask him. So as part of the ENCODE, we put together a registry of nearly 1 million CCREs in humans and less for mouse because we have less data for mouse. But having mouse and human enable us to start to think about conservation, which kind of led to what I'm going to tell you about, because this is kind of old news. You might have read about it, but for us, it's very old news, but it serves as a motivation. So if you take a look at these CCREs and you look at the Philo-P score. So we had in our hands at that time was a Philo-P score across 100 vertebrate genomes. And you can see these different types of CCREs. These are like promoter, enhancer, different. I'm not going to go into the details here. They are clearly more conserved than random, but they're actually not impressive. The reason I'm saying that is because I'm not showing you the error bar here. If I were to show you the error bar, they would be like huge, right? This is mean on average, they're more conserved, but a lot of them actually are not conserved. And if you compare the set we have between human and mouse, the human one is, I mean, it's a bit misleading here because the mouse set is smaller due to the smaller amount of data. But in any case, quite a bit of it is like no homology between human and mouse, and homology only, and then homology and also CCRE in the other species. So there is definitely conservation, but not so impressive. So for a while, we kept on pondering over this. Are these low Philo-P CCREs truly unconserved? Or the data we had for conservation was across 100 vertebrates. And it's a long evolutionary distance. Perhaps some of these CCREs will show conservation in more recent evolutionary time, right? So we got involved in this Zoonomia Consortium, which is the project that's led by Shishun Limbo and Eleanor Carlson. It just happens that Eleanor is one of my faculty. So we were talking about this during our faculty research, and she was like, oh, you're looking at this conservation. Why don't you try out our mammalian genomes? So they have sequenced 240 placental mammals. And that possibly is a more recent evolutionary time, right? We were like hit it off right away. And Greg was the one, Greg Andrews is a graduate student in my lab, was making the presentation. He must have done a very good job. So we got all roped into, this is what I'm going to talk to you about today, this project as part of the Zoonomia Consortium. So also Kylie Fan and again Henry, they are co-leading this manuscript. So we took the 200 mammal phylo P. So indeed, you see these solid lines, they are a lot higher than the dashed lines. The dashed lines are from 100 vertebrate in the previous plot I show you. So it's quite a bit, like 3 times higher. And we have our hopes up, so this is great. So another advantage of the Zoonomia data was it has these reference-free alignment made by an algorithm called the cactus. I mean, why would you use a plant to make the alignment of mammals is beyond me. But anyway, so they have across the 240 placental mammal alignment. Okay, it's not human specific. So which is better than the vertebrate one we had before. So we took the alignment, and so Kylie was the one who came up with this triangle representation. So if you take one CCRE and you look across all 240 species, okay, so anyway, it's a bit confusing. I don't know if I should get into it, but anyway, just remember it's 240 genomes, and they don't include the human in this because these CCREs are defined in humans. So by definition, they're already in humans. So we're interested in whether or not they are also aligned in the other genomes. And the reason that it's still 240 is because Eleanor is a dog person and it has 2 dog genomes in there, which is a bit jarring and like a purebred and some kind of cross. Anyway, so it's still 240, just a tidbit. So if you take a CCRE, remember it's like 300 base pairs long, and you can have some kind of threshold as to what percentage of the 300 base pairs need to be aligned in another genome. Okay, depends on the threshold. You can say 90% of it has to be aligned. Okay, and then you count across the 240 genomes, exactly out of 240, how many would be able to align more than 90% or more of the position. And you can also flip it and say, how many of the genomes really definitely don't align? So less than 10%. Less than 10% again of the 300 base pairs. All right. So by definition, every CCRE, this is 0.92 million dots on this heat map. Every CCRE needs to lie in this triangle because they can't go out of it just by design. Okay. But the interesting thing is 3 populations pop out. Group 1, we call them group 1, group 2, and group 3. And this is random. If you randomly pick the regions in the genome, you can see there is an enrichment in these 2 regions, especially group 1 and group 3. So as it turns out, group 1 is highly conserved across mammals, across almost 240 mammals. And group 3 is primate specific. Very primate specific, as you'll see in a minute. It's very interesting. And group 2 is a mix. I don't know if you have questions about it, I can show you more plots, but I just don't want to get into all these details without confusing you. Yeah. Oh, N1 is the number of species, number of genomes, sorry. N1 is number of genomes out of the 240 that can align at least 90% of the positions of a particular CCRE. N2 is at most 10%. So N1 and N2, some up to at most 240. That's why it's on the triangle. So you can see that group 1 is enriched. We're very enriched. So it's half of the CCREs are actually enriched across all the mammals. This is much more so than the vertebrate, which you will see in the next slide in comparison. So did I answer your question? I know it's a bit of mind twisting here. Okay, ask me again. Okay, so okay, we submit a paper and whatnot, and one of the reviewers was being very annoying at first, but turned out to be actually very prescient. He was like, "Why do you think mammals genome? Are you sure it's helping you? What do you get if you just use your vertebrate genomes?" So we actually went back and made this map with 100 vertebrate genomes. Now it goes to from 0 to 99. You don't really show it because human is part of the 100. And out of the 100 vertebrate genomes, there are 68 mammal genomes. Just keep it in mind. So you see the blob moved from here to here. Okay, and this region is the part that's conserved beyond the mammals. And if you really want to be very stickler about the ones that are conserved beyond mammals, it's this triangle at the top left corner. And that is only 4.4% of our CCREs. Okay, so basically you have to use a shorter evolutionary distance if you really want to detect the conservation of these elements. They evolve very fast. And if you go beyond mammals, you just don't detect them. You detect some really, really deeply conserved ones, but very low percentage. And you can take these map and then take each one, the CCREs in each region, and map to the mammals triangle. This is the same one as what I showed previously. And you can see the ones VG0, vertebrate group G, it's like at the pinnacle of this triangle. It's hugely conserved. So anyway, by using the mammals data, we really are at a very sweet spot for detecting conservation. So, okay, here comes another reviewer, which was really, really annoying. It was like, I mean, for a week, I was very annoyed by him, and I was losing my appetite because he says there's really nothing wrong with the paper, but I just don't like it. You know, how about you do this opposite thing? You know, you start with your alignment. You're not using the alignment. You're doing all this averaging. So, okay, we did something following the reviewers. Usually, you know, reviewers, they they don't help in a lot of the papers. It turned out to be, this one was really helpful. So we took the alignment. So each CCRE can be represented as an alignment, a percent alignment across all 240 genomes, right? And then we throw this thing into UMass. And if you still remember group 1, group 2, group 3 in that triangle heat map, and they show up, I was like, whoa, what is this? So group 1, remember, it's the across all mammals conserved. And then group 2 is the one at the lower left. It's a mixed bag. We call them actively evolving. And group 3 is primate specific. The reason it's primate specific, we have all the alignments. They effectively only show up in primates. So out of the 240 genomes, we have 42 primates plus 3 nearest non-primate genomes. They're very weird things. I actually learned a lot of biology about these things. The closest one is Thunder flying lemur, which is neither flying nor a lemur, right? And it's this naked red-looking thing with a canopy on top that can glide for like 250 meters from some tree. I don't know why you need to do that. But in any case, for like 50 years, people thought it was a mammal and just got recently disqualified. So anyway, all these tidbits aside, and these group 3 elements, they basically explode into little clusters, right? You can color this UMass by like number of genomes they align. And it's like fascinating, depends on what fraction of the length you require. You have heard the number of 90% and 10%, right? So if you color by 90%, like they align, the number of species that align is like at the tip here. But if you change the threshold to be a little more lenient, you see the red kind of crept up. So again, like one of the reviewers was asking, like how, like, you know, you're making all these arbitrary decisions, 90%, 10%. Like what happens if you change the threshold? There you go. A group 2 is actually this thing, it changes. This is green ones. When you change the threshold, it aligns more. So if you require 50% to be aligned, group 2 elements actually get aligned across like 170 genomes. So they really are a very interesting mix. And then of course we can start to color this thing by other possible genomic function like phylo-P score. So phylo-P score alignment is doesn't have like a mutation rate in it. So by design, phylo-P score has a mutation rate of the different branches of the tree already in the model. And so we think it's a more accurate model. You'll see in a minute when we switch to transcription factor binding sites. But you see the group 1, the most conserved one at the tip have the highest phylo-P score. And you can color it by the distance to the nearest transcription start site. You see these fins start to show up. Like even the most conserved ones, they don't have to be promoters, but promoters tend to be more conserved. And when you overlap with transposable elements, right, like the group 3 really are pretty much the majority of the ones that group 3 here are derived from transposable elements. So I'm going to switch to transcription factor binding sites just to show you related results. So as part of ENCODE, we have, Henry here again, built this resource called the Factorbook. You might have used before. Take the CHIP-seq data generated by ENCODE and then annotate as much as possible, motif, all kinds of things out of it. So we already, in our wheelhouse, we already have collections of transcription factor binding sites all curated. So we took 367 TFs, really the curated sites of these transcription factors. And now they are smaller. They're like between 7 to 20 base pairs big. So one CCRE can have a lot of transcription factor binding sites in it. Just keep that in mind. So it's a higher resolution picture of gene regulation. So this is one out of the 367 TFs we looked at. It's a liver-specific HNF4A transcription factor. And this is the corresponding UMass. Okay. Remember the one with CCRE, still again, red, green, and blue for the 3 groups. Red is highly conserved across all mammals. Green is the mixed cluster, mixed group. And then blue is primate specific. So we have these 42 primates plus like 3 other species. I mentioned flying lemur. The other 2 are tree shrews, in case you're interested. So here is the primate part of the tree. So the tree is longer. We just truncated it to show only the primate part. Okay, so you can see that all these clusters indicate different evolutionary combinations, like A here, it's only in grade 8. You don't have to be able to read it, but if you zoom in, you can see all the data with percentages and all that. But just look at the color. Red means it's there, and then blue means it's absent. If you look really carefully, it's not like 50%, 50%, it's more like 90% and 5%. So it's really clean cut. So this cluster is only in gray apes, and B, where did B go? Okay, B is only in gray apes and old world monkeys, and C is in gray apes and new world monkeys, but not in old world monkeys. And D is in gray apes and monkeys. Okay, E now extend to lemur-like monkeys, and F now includes those 3 interesting non-mammals. Okay, so you can look at each cluster. They are like each way you can imagine. Yes.
Binding sites is just a sequence level constellation of placental mammals.
Yes.
It's not from.
Oh, they're from yes. The data from humans is from CHIP-seq, but in other species, it's only sequence level conservation.
How do you know they're bounding now that they?
That's a really good question. I'll show some data for some of these. Okay, but they are, yeah, I mean, it's complicated. They may not be bound by the ones that have sequence conservation. We believe it's still bound in other species. Okay, just hold on your question. If I don't answer it, ask me again later. So if you look at these clusters again, if you look at the transposome element, you see these waves of transposable elements. So these are the same classes I'm talking about before. And like line 1 element here, line 1 is among the youngest transposable elements in our genome. Line 2 is old, very old. Alu is sign, is also very recent. Mir is sign, but ancient. And LTR is long terminal repeat containing element is young. And DNA element is old. So you can see like waves of them just bringing in the sites into the human, the primate lineage differently for the sites of one factor. And if this is not like HNF4A specific, if you look across like the 367 TFs we have data for each one of them, we have a heat map, we have all these things. You can see they basically tell the same story. And this is an aggregate plot across all the TF binding sites. There are 15.6 million of them defined in human. And you can see that if it's group 3, 86.1% of them are derived from transposable elements. And they are basically innovations of transposable elements during the primate lineage, and mostly contributed by these young Ts, line 1, LU, and the LTR elements. Because the resolution for transcription factor binding sites is much higher than for CCREs, which I showed you. If you still remember the plot before, the continuous portion, the portion that's not group 3, is quite continuous. But for transcription factors, this is more likely the case with like lobes coming off for other elements. And you can see this is FOXA1 binding sites. And these lobes of them, and you can see they're lost. Like 1 here is lost in bats, and 2 lost in New World monkeys. Again, red means present, blue means absent. 3 is this lobe was lost in cetaceans. These are whales and platypuses. Oh, okay. Sorry?
Time's out.
Oh, time's out. Okay. You can see the picture, and I'm gonna skip this, and then I can answer Jian's question. So at the end, okay. So here is CHIP-seq data from other species, and the ones that are this is the percentage of constrained sites. We actually came up with a way of defining subsets of them that are conserved across the species. And a subset of them, we think we know which ones are conserved, and they are likely to be bound and likely to be functional. And this is CHIP-seq data across species. And the constrained ones are likely to be bound. Okay, likely to leave footprints of DNA's cleavage. And at the end, I have to say this, but in order to get to my punchline, I do have a punchline besides color pictures. So we did LD score regression for all these sites. Here are the zoonomia conserved elements, and we derived a set of conserved elements using 2-component Gaussian model in one of the sites that I skipped over. So anyway, we defined the ones that we think are conserved with a better definition, using trees and mutation rate. And they are highly enriched in human traits. Okay, so the enrichment in the human trait is the most prominent when you have the highest conservation, and it's cell type specific for the traits. So we have thyroid traits, red blood cell traits, and then other immune traits. They correspond to different cell types. So I'm just going to conclude before I get thrown off the platform. So the conclusions, I have a very long conclusion, but anyway, I just want to tell you that what I really want to tell you is, so if you look at the human regulatory elements, about 1/5, like 20% of them are recent innovations in the primate lineage by transposons. Okay, however, that's not what's causing all our problems with our traits. So however, it's the opposite. The most deeply conserved elements across all mammals, which constitute about 1/8 of all of our elements, those are the ones that explain human traits. Okay, that's it. Yes.
Fascinating to see this. In the late 2000s, there was a lot of work from Eddie Rubin's lab on what kind of constraints, sequence constraints we use. Cis-Regulatory Elements. I contributed some of those papers that we not all. Okay. And so we published a lot of papers then showing that mammalian constraint, placental constraint, is what you should use for sensitivity detecting mammalian regulatory elements. And then in 2014, after we used a slightly different version of what you're doing, which is and so I was going to suggest that for your work as well. You're looking at conservation, how much is aligned, right?
Yeah.
But that doesn't tell you about constraints, which say phylo-P does.
Yeah.
You can essentially run phylo-P on different planes. You don't have to run phylo-P.
Yeah, this is basically what we did.
Exactly. So you look at constraint rather than, if you just look at conservation, then of course the primate specific elements will be transposons.
Correct.
Right?
Yeah.
But if you look at constraints, what you find is that the transposons are not all equal. So the vast majority of transposons, they are not constrained.
Correct. 99% of them are not constrained. 99.9% of the groups we are not constrained. It's exactly the same as we said.
Actually, much more. Actually, if you use primate alignments alone.
Oh, primate alignments alone.
Primate constraint is higher.
Yes. If you use primate alone, a lot of them align.
Exactly.
Yeah.
What I suggest is using primate. If you're looking at primate alignments with primate constraint, look at mammalian alignments with mammalian constraint. That gives you a better idea of the conserved function. And if you look at constraints, what it turns out is that transposons and non-transposons are the same. They're equally likely to give you functional elements. But you have to look at constraints in primates.
Yeah. Okay. We can look. So this is the slide I skipped over, and you can see the contribution. It's like a bimodal distribution. We did this for each of the TF, and we defined the other mode that's constrained.
This is primate constraint?
No, this is mammal constraint.
Right. Very different picture.
Yeah, we can do that. Yeah, we can do that for primate. They are like 43, 42 species. Yeah, we can repeat this for primates. Good suggestion. Thank you.
So remind me again, so the ENCODE data has about 680 transcription factors?
Yeah, so it has more than what I talk about. So basically here we showed 367 of them because we have curated a set with motifs, so we understand what they are. And ENCODE has more data for transcription factors.
As I recall, though, they're pretty depleted for the zinc finger class, which is less well studied.
Yeah.
Yet people are doing in vitro experiments to try to find binding motifs without those class.
Yeah, tin hues.
Right. So can you take that information and kind of feed that back into nucleosome occupancy data and other things from the ENCODE project to try to infer whether those sites are actually occupied?
They are occupied.
Yeah.
They are.
But trying to tie them to whatever transcription factor, like using the
we have, these are the crab zinc fingers. I skipped the slide. Yeah. So we have 69 of them in our collection, and we actually learned quite a bit about them. But they're bound. So they are definitely bound in humans. It's just they are recent inventions, so they're less likely to be found beyond primates.
Yeah. And, and they have more of a link to transposons, as I recall, because they're
exactly. Percent transposable element overlap with transposable elements. You see the bigger ones, they're all way high up. Yeah. And they have very different features too. They're more there to repress transposons, while others are being spread around by transposons. Yeah, that's right.
Yeah, so is that for now the code to associate CCREs to the target chips that are being related?
Yes.
So have you looked whether these different types of conserved elements are also conserved, the association between CCRE and
That's our next step. I was going to go to talk to Eris and use this DNA zoo data. It has high C data on a lot of mammals. We have not looked at them yet. Yeah, it's the next step.
So I thought that was really impressive, the explanatory power you got using explicit evolutionary trees and looking at the clades with the HMF4A binding sites. So I was wondering what you thought about looking at the analysis you did the first step, where you were taking a lump of all the 240 different mammals, and whether looking at those in the context of the details of the trees for that original analysis you were doing there.
We have like two sets of parallel analysis. One is for CCREs, which are bigger and lower resolution, and you can actually see they are more continuous. And then each one is like a combination. I haven't showed them to one. It's like it's a combination of different groups of transcription factor binding sites. So which kind of limited our analysis. That's why more analysis was done on TFESs. And that's why I show you more of it. But we have done both analyses. They are in the big picture, they are similar to each other. Functions are pretty much the same. Jen still have a question.
Yeah, is it okay if I ask?
Go ahead.
Yeah, of course, go ahead.
So the question is about, so you completely rely on the sequence alignment to call whether that position is conserved.
Yes.
But for these,
It's aligned.
yeah.
We call it
For example, these regulatory elements like the binding sites we show, we know there is some kind of
Turnover. They might jump to neighboring sites.
Yeah, the sequence level, the binding site may be conserved, but at the exact, alignment, the columns in alignment may not be that conserved. So
Yeah.
you go back to some of the elements that don't show the high of the, say, polyp conservation, maybe pick up some sequence level conservations.
It's possible. They have moved
by
around.
considering the
Yeah.
turnover events.
Yeah, it's
So in a
possible.
way that is at the sequence level is not that conserved, but functionally it is conserved.
It's possible. Yes. We didn't have the power to pin those down. I mean, we have some kind of way of allowing some movement in the alignment, but we didn't explicitly look for them in the neighborhood to see if they're still there. Yeah, that's a legitimate criticism.
Maybe it's time to thank Zhiping for a talk.
Thank you so much.
Outside for the photo session reception.